Good morning everyone. In this video we will see oxidation of fatty acids. Oxidation of fatty acids is usually called as beta-oxidation because the majority of the fatty acids in the body are oxidized on the beta carbon atom of the fatty acids. So that is, it may be defined, beta oxidation may be defined as the oxidation of fatty acids on the beta carbon atom, and this oxidation results in the sequential removal of a 2 carbon fragment, that is acetyl CoA. And these fatty acids are oxidized by most of the tissues in the body. However, brain, erythrocytes and adrenal medulla, they cannot utilize fatty acids for their energy requirement, and this oxidation takes place in mitochondrial matrix of the cell. Fatty acid synthesis takes place in the cytosol, but oxidation of fatty acids takes place in the mitochondrial matrix. That is, the essential enzymes which are required for the fatty acid oxidation are present in the mitochondrial matrix. And there are three steps that are involved in the fatty acid oxidation. One is activation of fatty acids occurring in the cytosol, and second one is transport of activated fatty acids into the mitochondria, and the third step is beta oxidation proper in the mitochondrial matrix. We will see one by one the reactions, these three steps. First one is fatty acid activation. This fatty acid activation takes place with the help of thiokinases, or these thiokinases are also known as acyl CoA synthetases. The reaction occurs in two steps and it requires ATP as well as coenzyme A and also magnesium ions. First step, if we see the fatty acid, this reacts with ATP, fatty acid reacts with ATP and forms acyl adenylate, and this acyl adenylate then combines with coenzyme A to produce acyl CoA, which is nothing but activated fatty acid. Here two high energy phosphates are utilized, and if we see here, ATP is converted to pyrophosphate, then the enzyme inorganic pyrophosphatase hydrolyzes this pyrophosphate to inorganic phosphate. So this is active, in that way the fatty acid gets activated to acyl CoA in the cytosol. This takes place with the help of thiokinases or acyl CoA synthetases. And there are three types of thiokinases are present to activate long chain fatty acids and medium chain fatty acids and short chain fatty acids. So this is the very first step of fatty acid activation. And second step is transport of fatty acyl CoA into the mitochondria. Acyl CoA is nothing but activated fatty acid. It should be transported to the mitochondria if it has to be oxidized. So as I told you, the fatty acid gets activated in the cytosol since fatty acid is synthesized in the cytosol. But if it has to be oxidized, then it has to move to mitochondrial matrix. But here the activated fatty acid, that is acyl CoA, is impermeable to the mitochondrial membrane. That's why a special carnitine carrier system operates here. That is nothing but carnitine shuttle. So, that is carnitine is nothing but a chemical, that is beta hydroxy gamma trimethyl amino butyrate. Here what happens in the cytosol is activated fatty acid, that is acyl CoA, combines with the carnitine with the help of an enzyme carnitine acyl transferase 1, so that it forms acyl carnitine and then coenzyme A. And here this acyl carnitine, now it is freely permeable across the mitochondrial membrane through a carrier protein system, and it enters the mitochondrial matrix, and here it entered mitochondrial matrix acyl carnitine, and here it combines it is going to be split into carnitine and acyl CoA with the help of coenzyme A, and then carnitine acyl transferase 2. So, here carnitine is again released and activated fatty acid is also formed in the mitochondrial matrix. Whatever the carnitine that is released, that is again formed in the mitochondrial matrix, freely permeable across the membrane, mitochondrial membrane, and enters the cytosol. There it again it is reused for the activation of another fatty acid. And about acyl CoA, which is nothing but a activated fatty acid, so it participates in the beta oxidation of fatty acids. And third step is beta oxidation proper. So, now we have activated fatty acid in the mitochondrial matrix, and this is going to be oxidized in 4 steps, that is activated fatty acid, that is acyl CoA is there. So, this undergoes oxidation reaction with the help of acyl CoA dehydrogenase enzyme, which is a FAD dependent enzyme, and forms trans-enol CoA. Here FADH2 are formed, which then enters the electron transport chain, and they are going to form ATPs. So, here if you observe with the formation of trans-enol CoA, the double bond is formed between the two beta alpha and beta carbons, and this is the first reaction of beta oxidation proper. And second reaction of beta oxidation is hydration reaction. Hydration with the trans-enol CoA with the help of enol CoA hydratase enzyme leads to the formation of beta hydroxy acyl CoA, and this is going to be catalyzed by the beta hydroxy acyl CoA dehydrogenase enzyme, which is NAD dependent enzyme. And here NADH are produced. These NADH again enters the electron transport chain, and they are going to form ATPs. So, this is also another oxidation reaction. And fourth step is cleavage reaction. Whatever the beta keto acyl CoA is formed during the oxidation of beta hydroxy acyl CoA is going to be cleaved with the help of thiolase enzymes into acyl CoA and then acetyl CoA. Here what is if you observe this acyl CoA, which is formed as the product of beta oxidation proper, will consist of two carbons less than the original carbon which participated in the beta oxidation. So, the initially we have the activated fatty acid with 4 carbons, 1, 2, 3, 4 carbons, whereas the final acyl CoA which is formed in the beta oxidation consists of only 2 carbons. That is, 2 carbon fragment is released as acetyl CoA and the remaining 2 carbon compound is acyl CoA. So, that means 2 carbons, the acyl CoA is formed with the 2 carbons less than the original carbon, original fatty acid. So, this acetyl CoA whatever formed or whatever released as the product of the beta oxidation will participate in the Krebs cycle and further it is going to be oxidized. And how about this acyl CoA which is formed as the product and which is the product of beta oxidation again will participate in the beta oxidation proper. So, that is it depends, this step depends on the length of the chain. Until the fatty acid gets completely oxidized, so this acyl CoA will be oxidized through beta oxidation proper. In that way, the beta oxidation takes place. So, this is regarding oxidation of even carbon fatty acids. Now, we will see the oxidation of odd carbon chain fatty acids. Odd carbon chain fatty acids, how it takes place is the beta oxidation of saturated fatty acids, of course, containing odd number of carbon atoms will takes place in the same manner like even carbon fatty acids, but the only difference is that the final product that is formed is propionyl CoA, which is a 3 carbon compound, and this has to be converted to succinyl CoA, and that succinyl CoA ultimately will enter the TCA cycle and further gets oxidized. But how does it takes place? This propionyl CoA, it is a 3 carbon compound. With the help of some certain enzymes, this is going to be formed or this is going to be oxidized to succinyl CoA. We will see what are those enzymes and what are those reactions. So, this propionyl CoA it is going to be carboxylated in the presence of ATP, then CO2 and vitamin biotin, and it will form dextroform of methyl malonyl CoA. So, this methyl malonyl CoA with the help of methyl malonyl CoA racemase, it will be converted to L-methyl malonyl CoA. So, D form is converted to L form, and this step is essential for the entry of this compound into metabolic reactions of the body. D form must be converted to L form for further reactions or for further metabolic reactions of the body. And the next enzyme is methyl malonyl CoA mutase, and this enzyme is dependent on B12, and with the help of this enzyme, this methyl malonyl CoA gets converted to succinyl CoA. This succinyl CoA enters the TCA cycle and further gets oxidized. And what about this if in the case of deficiency of vitamin B12, So this methyl malonyl CoA cannot be converted to succinyl CoA. If methyl malonyl CoA, L-methyl malonyl CoA is not going to be converted to succinyl CoA, it is going to be accumulated in the tissues and it is going to form methyl malonic acid. So this methyl malonic acid leads to methyl malonic acidemia. So this methyl malonic acidemia is nothing but due to the deficiency of vitamin B12, or it may be due to defect in the enzyme methyl malonyl CoA mutase. And if we see the symptoms of this, or what is going to happen, this methyl malonic acid accumulates in the tissues. It increased excretion of urine we can observe in these patients, and this causes severe metabolic acidosis, and sometimes it damages the central nervous system and retards the growth, and it is often fatal in early years of life. So this is regarding methyl malonic acidemia, which happens due to the deficiency of vitamin B12 and also defect in the enzyme methyl malonyl CoA mutase. And another disorder which I would like to talk here is sudden infant death syndrome. The exact cause of the sudden infant death syndrome is not known, but 10% of the SIDS is due to the deficiency of acyl CoA dehydrogenase, deficiency of medium chain acyl CoA dehydrogenase, and this is the step we found. The first step of β-Oxidation proper. Activated fatty acid undergoes to form trans-enol CoA with the help of acyl CoA dehydrogenase, which is a FAD dependent enzyme. If this acyl CoA dehydrogenase enzyme deficiency is there, what is going to happen is, we know in infants glucose is the principal source of energy. That is, after feeding or after eating, after eating or after feeding of few hours, these glucose levels decreases, or the glucose utilization decreases. When glucose utilization decreases, the rate of fatty acid oxidation simultaneously it must increase to meet the energy demand. But in case of SIDS, due to the deficiency of acyl CoA dehydrogenase, the blockade in β-oxidation occurs. Suddenly the death may happen to the infants. And another important disease that will happen in oxidation of because of the abnormalities of oxidation of fatty acids is Jamaican vomiting sickness. So this takes place by eating unripened ackee fruit, which consists of unusual toxic amino acid hypoglycine. This toxic amino acid hypoglycine, it inhibits the enzyme acyl CoA dehydrogenase, so that it is also going to block the β-oxidation of fatty acids, and it leads to various complications, as we have seen in the earlier case also. So that is regarding Jamaican vomiting sickness. So this is all about oxidation of fatty acids, especially saturated fatty acids with even number of carbons and saturated fatty acids with odd number of carbon atoms. Thank you.